And we'll go with Rodrigo that is going to talk about sensors on cotton.
Good morning. I'm an agronomist from Federal University of Mato Grosso, and I'm here now with my master's degree advisor by Professor Molin. And I'm here to talk about crop canopy sensors in cotton. So cotton is a very important crop in Brazil. This year we are cultivating 1.12 million hectares of these. These are the more important production regions, Mato Grosso and Midwest Bahia. And one characteristic of this crop is that farmers tend to use large areas. So we have farms from 500 to 20,000 hectares. In each field we have from 800 to 500 hectares. And some people, they work all these at the same, and in some circumstances all the farm are the same practice. Cotton is a perennial plant cultivated as an annual crop, and it has some implications. It has a long growing period from 150 days to 200 days, and the plants tend to keep growing, which can sometimes become excessive, so it's a problem. If you have too much vegetable growing, we don't have too much yield. Like in these situations, we have also problems with diseases. So there is a need to control the crop growth, and to do that we use plant growth regulators. And they change the hormonal balance of the plant, allowing better partitioning of photostimulates between vegetative and reproductive development. And we know this varies a lot, so we have lots of variations in plants, so it makes sense to use the right amount in each of the plants. And cotton is also a high fertility demanding crop, so it needs a high base saturation soil, no tolerance of acidity. So it's the crop needs a good soil to grow. And we know that adequate dose of nitrogen increases yield and improves fiber quality. But in excess, nitrogen can cause an imbalance and greater vegetative growth rather than reproductive development, which is also a problem. And it also increases the risks associated with pests and diseases. Like in this picture, we see where they apply too much nitrogen and we have problems with insects and then fungi. So as I said, they tend to use large fields, large farms, and we know some of them has high spatial variability of soil and also crop conditions, and they tend to treat it all as the same by the mean, as an average. So we know there is a big potential for variable rate application technology, which has already been done from lime and fertilizers, potassium, phosphorus based on grid soil sampling, but also potential for variable rate application based on canopy structure of nitrogen and plant growth regulators. What are the main inputs in this program? So the main objectives of this work we are doing here, it's a big project. The first objective is to evaluate the performance of active crop canopy sensors. We are using the ESALQ sensor, crop circle, crop spec, and also an ultrasonic sensor to measure crop height, and to evaluate their performance in predicting biomass accumulation and nitrogen uptake in cotton. So this is the first step, to do the calibration to understand how to relate the sensor readings to the amount of nitrogen and biomass the crop has already taken. The second objective is to establish the algorithms for variable rate real-time nitrogen application based on sensor readings. So we are doing some field comparisons, some sampling there, and then we can establish this relation between the sensor readings and the value the crop has actually uptaken, and then we need to establish these algorithms to establish how much nitrogen to apply in each situation. And talking with you about these algorithms, as Lucas already told something about sugarcane, we need to test which strategy is best. So the three objectives to evaluate the best strategy for redistributing fertilizer according to the spatial variability of crop detected by sensors. And I put these two pictures here are the, like he said, the two main ideas. One that says, so if the sensor readings are low, the plant needs more nitrogen. The nitrogen is the crop. And the other that says, no, if the sensor readings are higher, then the crop will, the yields will be greater, so we need more nitrogen, and we are testing that. And we are also thinking about quadratic forms and other ways to redistribute that nitrogen. And to be able to answer that question, we are also looking at evaluating the site-specifically the response of cotton to topographic nitrogen and the economic optimal rate of fertilizer. So we are using some classical plots with different doses, trying to use different management zones. So in a low potential situation, what is the best rate to apply, and in a higher yield potential situation, what is the right amount to apply. And lastly, the fifth one is to evaluate the potential of using variable rate applications of plant growth regulators according to crop vigor. So you know, these are crops in the same field at the same date, and we see lots of variability. These plants are a lot taller than this one, so probably they need more plant growth regulators to keep it in the right height to be able to harvest and other crops. And so that's the last objective. For this is a project of initially think of to be 3 years. The first one was just the relation between sensor readings and crop characteristics. The second one, which was this year, we also evaluated the algorithms, and we did it in this farm in Chapadão do Céu, state of Goiás. Here, one of the municipalities that has representative cotton, and here we are, so it's 800 kilometers from here. This is a picture of our research location, so it's mostly in flat areas, really flat, like 50 kilometers flat, and this is the situation. We had beans planted here, then they harvested the beans and planted the corn in January. And all these objectives, we are also considering the possibility that there is some interaction of these two main systems used in cotton. So, what are these? The conventional one, which uses row spacing about 80.80 meters, 80 centimeters, and it uses a population of 80,000 to 100,000 plants per hectare, and usually it is the row spacing used in the early plants. So planting done in December tend to use this spacing, but also some in January. And in this situation, the previous crop can be a cover crop or beans or even a short time soybeans. In the narrow row system, we have like half of the spacing, so from 40 to 50 centimeters, and we have almost double the population, so from 130,000 to 200,000 plants per hectare, usually after soybeans and planted in January to February. This is the dates of our field sampling and data acquisition. So we use these 2 dates mostly thinking about nitrogen applications. This is the dates they usually do the top dressing. And from these to even these, they use the plant growth regulators. So we are also evaluating this. And in the late season, we are trying to evaluate the potential of the foliants and openers in cotton. And this is the hardest to see how it yields. So this is what we are measuring: fresh matter, crop height, dry matter, nutrients, in all these 3. And then in this just fresh matter and crop height, again fresh matter, crop height, the foliation level and the percentage of open bulbs. So this is a picture of these 2 sensors. We are using 4 in total. These 2 I showed at last conference here. So this is the principle. They emit sunlight, they receive it, they calculate a vegetation index. We relate that to the amount of nitrogen and biomass the crop has uptaken, and then we, in real time, we determine what is the right amount of nitrogen to be applied. After we go with the machine in the fields and we got the sensor readings, we use the map to establish some sample points. We are using, in this situation, 30 sampling points, and we choose some places that better represent the variability of the field. So like this, some points where we have low biomass, some points where we have the higher biomass, intermediate one, and so on. And in each point, we hand measure the crop height to compare with the ultrasonic readings and also the other sensors. And we cut all the plants in 3 rows, 1 meter long each one. We weight it to know the biomass accumulation. Then we send it to the laboratory and we analyze the dry matter, the egg content, and also other macronutrients. And these are some first results. So this research is still going on. We are in the middle of it, and this is our first results. So we have seen good correlations between crop height and fresh matter in cotton. There was a significant interaction between the different narrow row systems, the row systems, the narrow and the conventional one. So we need to do for each one equation, but in both there were good results. We can see in the conventional one, we have in the beginning we have already less biomass, because here we have a lot more of plants. The plant population is almost doubled, but it tends to increase faster because the plants have more space and there is more rain because it's planted earlier than in this other system. So we know that we have always to consider which system is and to make different recommendations in each one of them. And the same holds true to nitrogen uptake. So we have here R squared of about 0.90, 0.91. It's a really good correlation between crop height and nitrogen uptake in both systems. And here are the last results we presented in the Brazilian Conference of Precision Agriculture, comparing these 2 sensors. like you said, we're seeing that these used to spreaders, they have a large footprint, so they have been better than the other ones, although we didn't do all the analysis yet. And this is the correlations we have between the any sensor readings and the fresh matter. And here we consider, again, we had an interaction between different systems, the row spacing, but we already considered it and we put in the graphs the predicted value and the observed value considered this interaction. And we had really good correlations. The R squared here about 0.96, 0.92 from cop spec. So the sensor showed a bit better, but both can be used, both have good correlations. And also with nitrogen uptake, it's almost the same thing. Some noise here in some situations, but in general it's a good prediction. So what we have seen until now is that these crop sensors are good to predict biomass or nitrogen accumulation in cotton in both systems, but the equations should be different. They are different. And that's it. I thank you for your attention. If you have any questions,